Hey, welcome to the first video. In the next 8 minutes, we find out what radio-HPLC really is. Let's start. Every day, in hospitals and research centers around the world, patients receive radioactive drugs. Before any radioactive drug reaches a patient or enters a research protocol, it must pass a series of quality control tests. One of the most important of these tests is performed using a technique called radio-HPLC. In this video, you will learn what the radio-HPLC is, where it fits in the radiopharmacy workflow and why it's fundamentally different from the normal HPLC, which you may already know from classical analytical chemistry. Let's start with a brief recap what HPLC is, because radio-HPLC builds directly on this foundation. If you are new to HPLC in general, I recommend that you first watch Module 1 of the HPLC-Academy, where all of these concepts are covered in full detail. Here we will keep it brief. HPLC stands for High Performance Liquid Chromatography. It is a separation technique in which a liquid sample is pushed through a column packed with small particles under high pressure. Different compounds in the sample interact differently with the stationary phase inside the column, which causes them to travel through the column at different speeds. As they exit the column one after another, a detector records a signal and the result is a chromatogram, a graph with signal intensity over time. Each peak in a chromatogram represents a compound. The position of the peak is called retention time. It tells you something about the compound's identity. The area under the peak tells you something about the quantity. In classic HPLC, the detector is typically a UV detector measuring how much ultraviolet light a compound absorbs as it passes through the flow cell. Other detectors are fluorescence, refractive index, conductivity, and mass spectrometry-based detectors. They are all covered in Module 1 of the HPLC-Academy. This well-established technology used in pharmaceutical quality control, food testing, environmental analysis, and many other fields. In radiopharmacy, we take the same platform and extend it with one additional specialized detector, the radio-detector. Let's have a brief look what a radiopharmaceutical is. Radiopharmaceuticals are drugs in which one or more atoms of the molecule have been replaced with or attached to a radioactive isotope, also called a radionuclide. They serve three major purposes: diagnostic, therapy, and research. Across all this application, a key analytical challenge is to ensure the purity of the drug and detecting unwanted byproduct, and that the radionuclide is correctly bound to the drug molecule. With this background in mind, let us look at the core analytical challenge. When you analyze a radiopharmaceutical with a standard HPLC, the UV detector tells you about the chemical composition of the sample, peak for your product, unreacted precursor, and other UV active species. But it cannot distinguish between the radioactive molecule and the non-radioactive molecule. In radiopharmacy, this distinction is critical. Considering a sample of 177 lutetium PSMA, it may contain the correct labeled product, an unlabeled PSMA ligand, and free lutetium ion still in the solution. The UV detector cannot tell you how much radioactivity is in the fraction. Standard HPLC alone cannot answer the essential quality control question: what fraction of the radioactivity is in the correct chemical form? To answer that, you need a detector sensitive to radioactivity decay, the radio-detector, right? A radio-HPLC system is a standard HPLC system with an additional radio-detector connected downstream of the conventional detector. The conventional detector, typically UV, is called the cold detector, and the radio-detector is simply called the radio-detector or hot detector. The flow path is straightforward. First, the pump delivers the mobile phase at a constant flow rate. Second, the sample is injected and entering the column. Third, the column separates the components of the sample based on their chemical properties. Fourth, the element flows first through the cold detector, which records the UV and other chemical signals. Fifth, then it flows continuous to the radio detector, which records the radioactivity signal. Sixth, both signals are recorded simultaneously resulting in 2 chromatogram from a single injection. This combination is the analytical foundation of radiopharmaceutical quality control. Now, where does radio-HPLC fit in the overall QC workflow? Radiopharmaceuticals production follows a strict sequence: synthesis, purification, formulation, and then quality control before patient release. The QC test panel typically includes appearance, pH, radionuclide identity and purity, radiochemical purity, chemical purity, residual solvent, sterility, and bacterial endotoxin. Before we close this instruction section, it is important to address a concept that will be explored in full detail in section 4 and section 5 of this module. It's radiochemical purity and the critical difference between radio-TLC and radio-HPLC. Radiochemical purity, or RCP, is the fraction of the total radioactivity in your sample that is associated with your intended compound in the correct chemical form. Both radio-TLC and radio-HPLC are used to measure RCP, but they do not measure the same thing. And they do not give you the same information. That's important. Radio-TLC is excellent at detecting free unchelated radionuclides and highly polar radioactive species. A free metal ion such as free gallium or lutetium is highly polar and will stay at the origin of the TLC plate or migrate very differently from the labeled product. Radio-TLC captures all radioactivity on a plate, so none is lost. This is the great strength of this method. However, radio-HPLC has a known weakness here. Free radionuclides and highly polar species often absorb strongly to the HPLC column, the system, the tubing or injector, and simply do not elute. They are invisible to the radiochromatograph. If you rely only on radio-HPLC to detect free radionuclide, you may miss it entirely and report a falsely high RCP. On the other hand, radio-TLC has its own important weakness. It cannot resolve closely related radioactive impurities. Consider a labeled byproduct that is structurally similar to your product. For example, a radiolabeled synthesis side product or radiolysis degradation product. These species have nearly the same polarity as your product and will co-migrate on the TLC plate, hiding under the large product peak. They are simply overpowered and invisible. Radio-HPLC, with its far superior chromatographic resolution, can separate these impurities when the method is appropriately designed. A correctly developed radio-HPLC method can resolve labeled synthesis byproducts, radiolysis degradation products, enantiomeric or diastereomeric form of the labeled compound when you use the special column, and side products from competing reaction during the labeling process. So the conclusion is here: the two techniques are complementary, not interchangeable. Radio-TLC is the method of choice for detecting free radionuclides and providing a complete account of the total radioactivity distribution. Radio-HPLC is the method of choice for resolving and quantifying labeled impurities. In section 5 we will go through this comparison in full detail with concrete examples. Let us summarize what we covered in this first section. Radiopharmaceuticals are used for diagnostics, therapy and research. They contain radionuclides that span a broad range: PET and SPECT isotopes for imaging and beta and alpha emitters for therapy. Standard HPLC alone is not sufficient for radiopharmaceutical quality control because it cannot detect radioactivity and distinguish between radioactive and non-radioactive molecules. The radio-detector added to the HPLC system provides this capability. Radio-HPLC and radio-TLC are complementary techniques. Radio-TLC is best for detecting free unbound radionuclides. Radio-HPLC is best for resolving labeled impurities and providing simultaneously chemical and radiochemical purity information. Neither technique alone gives the complete picture. In the next section, we will have a look at both detectors in details, the cold detector and the radio detector, and what different radio detectors they are, how they work, and how to choose the right one for your compound. Stay tuned for the next video.